You know, it's kind of wild when you think about it. Feline leukemia virus, it's incredibly common,
Yeah,
but it's also like a really serious threat to cats, everywhere in the world.
absolutely.
I mean, we're talking about a virus that can lead to like so many problems from, like swollen lymph nodes
Mhm.
to just being constantly tired to like the really scary stuff like anemia and even leukemia itself.
It's true. It's a huge concern in feline health,
Yeah.
and to really get a handle on it, we need to like dig deeper into this virus.
Yeah, for sure. And that is exactly what we are doing today.
I love it.
So we've got some really cool new research that's kind of like looking at how we classify FeLV in a whole new way.
Okay.
And it turns out that the way we've been doing it, like the traditional methods, may not be giving us like the full picture.
Okay.
And this new perspective, it could tell us a lot more about the virus itself and, where it pops up in different places around the world.
Yeah, that's really important. And we're going to look at a study that actually goes into the genetic makeup of FeLV to understand it on a much deeper level.
Okay.
And we also have this really interesting case study uh, about a really unusual case of erythroleukemia,
Okay.
which is a kind of blood cancer, a really aggressive one
Yeah.
that was actually linked to an FLV infection. And then we've got a survey that gives us like a glimpse into how common FLV and FIV are among like healthy cats in Thailand.
Wow. Thailand specifically.
Yeah, so we're going from like the tiny molecular level to like what's actually happening out there in the world.
I love it. I love how we're covering the whole spectrum. So, okay, big picture here. By the time we're done with this deep dive, what should they walk away with? Like, what are the key things they should know?
So you're going to have a much clearer picture of like the different ways we can categorize FLV like beyond the standard tests that we do.
Yeah, okay.
We're also going to look at some really interesting ideas about how the virus itself might be evolving.
Okay.
And we're going to take a look at its presence, in Thailand in this case. And the goal is to give you guys those aha moments that help things really click without, getting too long in the weeds.
Without being overwhelmed, right?
Exactly.
Okay, perfect. So let's start unpacking all of this. So when we talk about like the current way we classify FeLV, like what is the go-to method that's being used right now?
So right now we mainly use something called phenotypic testing.
Phenotypic testing.
Yeah, and it's basically like interference assays.
Okay.
And this helps us identify all these different subgroups of the virus,
Okay.
like FeLV A, FeLV B, CE, and DCE.
So there's a whole bunch of them.
Yeah, there's a bunch of them. And the way these tests work is by basically watching how one virus messes with another virus's ability to replicate,
Oh, wow.
or by figuring out which specific receptors on a cat's cells that the virus uses to get in.
So it's like observing their behavior in the lab.
Exactly.
Okay. But this new research that you mentioned, this is saying we should be looking at something else, right?
Yeah, so this new approach looks at the genetic code of the virus.
Okay.
And specifically, it's looking at a part of its outer layer called the SU gene.
The SU gene.
Yeah, it stands for surface envelope. And think of the SU protein like it's a key on the virus.
Oh, okay.
And it has to fit into a specific lock on the cat's cell to get inside.
I see.
And any changes to this key can make it fit different locks or like fit better or worse.
That's a great analogy.
Thanks.
So how much information did they actually analyze for this new classification method? Was it like a small sample size or did they have a lot of data to work with?
So they actually looked at a ton of data.
Really?
They analyzed 404 publicly available SU gene sequences.
Wow. That's impressive.
Yeah. And the really cool part is that for 63 of those sequences, they also had the results from the traditional phenotypic test that we talked about.
Oh, okay.
So they could compare like the old way and the new way, side by side.
That's really smart. That's really good design. So what did they actually find? Like when they did this genetic analysis, did it show the same groupings of the virus as like the old phenotypic tests?
Not exactly.
Yeah.
So the genetic analysis pointed to two main genetic clusters,
Okay.
or clades, as they're called.
Clades.
Yeah. And one of them they called the FeLV-A clade.
Okay.
And this group had sequences that we traditionally call FeLV-A, but also FeLV-CE and FeLV-T.
Oh, so there's grouping some different ones together.
Yeah, and the other main cluster they call the NFeLV clade.
Okay.
And this one brought together FeLV-B and what we call endogenous LV or NFeLV strains.
Endogenous FeLV, what does that mean?
So endogenous retroviruses, like in FeLV, they're like viral sequences that have become a part of the host's own genome
Oh, wow.
over like a really long evolutionary time.
So it's like integrated into the cat's DNA.
Exactly. And they usually don't actively produce new viruses,
Yeah, okay.
but they're still there in the cat's DNA and they can be passed down to their kittens.
That's so interesting. So basically, this new genetic clustering is suggesting that the relationships between these FeLV subgroups might be different than what we originally thought.
Exactly. So they're proposing that FeLV, BCE, and FeLT, they all probably came from FeLV.
Okay.
And this happened through either small changes in their genetic code, which we call mutations,
Right.
or through a process called recombination.
Recombination, so like a swapping of genetic material.
Exactly. So they think that FeLVB and NICT, they probably came about from this mixing of genetic material between FeLVA and those NVLV sequences that we were talking about.
Wow.
And then FeLVT, that one seems to have evolved from FeLVA through like a combo of deletions and insertions. So like losing or gaining little bits of genetic code.
So it's like these more potentially harmful subgroups, they might have actually evolved by picking up genetic bits from not just other viruses, but from the cat's own genome.
Yeah, it's a really fascinating possibility.
That is mind-blowing.
But they did say that they had some trouble classifying FeLVE genetically in this study because they didn't have enough sequences for that one.
Oh, I see. So, but this whole idea of recombination, you said it is a major player in how FeLV evolves. Can you talk a little bit more about why that's so important?
Sure, yeah. So when a cat gets infected with like an active FeLV, like FeLAVA,
Ah.
that virus, it can meet up with and interact with these NLVL sequences that are already in the cat cells.
Okay.
And when the virus is making copies of itself, that's when the genetic material from the active virus and that endogenous sequence can get swapped.
Oh, wow.
And this recombination with NLVLV, it can lead to like brand new FeLV variants.
Oh, wow.
And sometimes these new combos, they can end up being more pathogenic.
Meaning they're more likely to cause serious problems.
Exactly.
So you mentioned earlier is that FeLV and NMLV are actually pretty similar genetically.
Yeah, so when you look at their nucleotides, which are like the building blocks of DNA and RNA,
Okay.
they share about 86% similarity.
It's a lot.
Yeah, and that's part of why these recombination events are more likely to happen.
I see. So let's talk about the SU protein again. What exactly does it do? Like, what's its role?
So the SU protein, that's the surface envelope protein we talked about, it's like the
Key.
that the virus needs to get into a host cell.
Got it.
It's the part that actually binds to receptors on the cat cells. So any variations in that SU protein can change which types of cells the virus can infect.
Okay.
That's called cell tropism. And ultimately, it can affect how the virus makes the cat sick, how pathogenic it is.
So these genetic changes in the SU gene, they're super important for understanding how FeLV adapts and interacts with cats.
Exactly.
Okay, cool. So the researchers, they also found these variable regions in the SU gene.
Yes.
What are those and why are they important?
So they found these 5 spots in the SU gene that have a lot of variation in their genetic sequences,
Okay.
and they labeled them VRA, VRB, VRC, VRD, and VRE.
It's like a whole alphabet soup in there.
It is.
So what did they see when they looked at these variable regions in those 2 main clades?
So they found these patterns of insertions and deletions in each clade.
Okay.
So insertions are like adding in a bit of genetic code, and deletions are taking some out.
Okay.
So for instance, in the FeLVA clade, they usually had insertions in the VRA and VRC regions, and a deletion in the VRB region. But the NVLAV clade was different. They typically had deletions in VRA and VRC, but an insertion in VRD.
So it's like each group has its own unique genetic fingerprint.
That's a great way to put it. And these differences probably mean that the viruses function differently too.
Like they might affect which receptors the virus can bind to or how well it can get into cells and start replicating.
Exactly.
That's fascinating. So they also looked at where these different FeLV types are found geographically.
Well, they did.
Did they notice any patterns there?
They found some clues about geographical clustering within that FeLVA clade.
Okay.
So the sequences from Japan, they tended to group together, and they were distinct from the ones found in Brazil, the USA, and the UK.
Wow.
And they also saw that the non-recombinant FeLV strains were mostly from Japan and China.
So it does seem like there might be certain strains of FeLV that are more common in certain parts of the world.
Yeah, that's definitely a possibility, but the researchers were careful to say that they didn't have enough global data to make any really strong conclusions about the link between geography and the virus's evolutionary history.
Okay, so they need more data to really be sure.
Yeah, a much bigger global survey would be needed.
Makes sense. So we've got this new and maybe better way to classify FeLV and understand how it's evolved. But you also mentioned a case report.
Yeah.
How does that fit into all of this?
So this case report is a close look at a very specific and rare situation.
Okay.
It's about a cat that developed acute erythroleukemia, which is a very aggressive cancer that affects the cells that make red blood cells.
Oh, wow.
And this cat was also infected with FeLV.
Oh, no, that sounds really serious.
Yeah.
What kind of symptoms did this cat have?
So the cat had a bunch of really worrying symptoms like loss of appetite, fever lethargy,
Oh, gosh.
and really severe anemia, and something called rubercytosis, which means there were immature red blood cells in the bloodstream.
Okay. And they were able to confirm the FLV infection.
Yeah. So the cat tested positive for the FeLV antigen using an ELISA test,
Okay.
which usually means there's an active infection.
All right.
But then they did a PCR test, which looks for the virus's genetic material, and that one was negative.
Oh, that's strange.
Yeah. So this can sometimes happen in what are called regressive infections.
Regressive infections.
Yeah. So in these cases, the cat's immune system might have suppressed the virus.
Okay.
So it's still there, maybe hiding out in the bone marrow or other tissues.
Okay.
But it's not shedding enough to be picked up by a PCR test on the blood.
So it's like the virus is laying low.
Exactly.
Okay, so they suspected this rare leukemia along with a possible FeLV infection. What did they do to treat the cat?
They tried high-dose cytarabine chemotherapy.
Okay.
And cytarabine is a chemo drug that's often used in treating acute myeloid leukemia in people.
Okay.
So in this case, they gave it to the cat through a continuous infusion for 12 hours each day for 5 days in a row.
Wow, so pretty intense.
Yeah.
So what happened?
At first, it seemed like things were getting better. The cat's symptoms improved a bit, and the anemia got a little less severe.
Okay.
But the other abnormal blood results, they didn't really change much.
Oh.
And sadly, the cat ended up dying from what looked like shock about 2 weeks after they gave a second round of cytarabine.
Oh, no, I'm so sorry.
Yeah, it's tough.
So how long did the cat survive after the diagnosis?
67 days.
That's heartbreaking. Did they learn anything from the post-mortem exam?
Yeah, so they looked at the bone marrow,
Okay.
and they found a really low ratio of myeloid cells to erythroid cells.
Meaning?
It means there was a massive overgrowth of those red blood cell precursors.
Wow.
In fact, those erythroid progenitor cells, they made up 64% of all the cells in the bone marrow.
That's a lot.
Yeah, and then the blast cells, which are those immature cancer cells, they were 84% of the non-erythroid cells. And when they looked at the spleen under the microscope, it was enlarged because it was packed with these atypical round cells, which were probably those erythroid precursor cells.
So everything pointed to acute erythroleukemia in a cat that also had FeLV. And the people who read the case report, they said this was the first time anyone had used high-dose cytarabine for this specific condition in a cat with LV.
That's right.
So what did they think? Did it work?
They said that it did seem to help a little bit at first with the symptoms and the anemia,
Okay.
but ultimately it didn't lead to a long-term remission or help the cat survive.
Right.
And they emphasized that erythroleukemia, it is linked to LV infection.
Yeah.
And unfortunately, it usually has a really, really bad prognosis in cats.
It's just so hard to hear.
It is.
But it's important information to know. So they also stress that we need more research to find better chemo protocols for this kind of cancer in cats.
Absolutely.
All right, so let's move on to that survey that was done in Thailand. What was the goal of that study?
So they wanted to see how common both FIV and FeLV are in healthy outdoor cats in Thailand.
Okay, so not just cats that are already sick.
Yeah, exactly. And they looked at different areas within the country, the northeast and the central regions.
Okay, so a pretty good representation of the country. So how many cats did they include in this survey?
They had 260 healthy cats from 13 different locations across those 3 regions.
All right, and how did they actually test these cats for FeLV and FeLV?
They used a rapid ELISA test.
The ELISA test.
Yeah, it's the same kind of antigen test that was positive in that erythroleukemia case we talked about.
Okay.
And for FeLV, they used a similar rapid antibody test, which looks for the cat's immune response to the virus.
So what were the overall results? Like, how common were these viruses in these healthy cats?
So overall, 5.8% of the cats had FIV antibodies, and 4.2% had the FeLV antigen.
Okay.
And only a tiny fraction, like 0.4%, tested positive for both viruses.
Those numbers seem kind of low, especially when you hear all the scary stuff about these viruses.
Yeah, they are lower than some of the rates that have been reported in other studies, especially ones that included sick cats.
Right.
And the researchers pointed out that they did find some differences between regions.
Like what?
So it looked like the prevalence of the FeLV antigen was a bit higher in certain areas like Lamphun in the north and in Bangkok.
Oh, interesting. So did they look into any risk factors that might make a cat more likely to be infected?
Yeah, they did a risk factor analysis.
Okay.
And they found that older cats that were 2 years or older, they were more likely to test positive for FIV antibodies.
Which makes sense because older cats probably have had more chances to be exposed.
Exactly, like through bites from other cats.
Right. But they didn't find any clear risk factors for FeLV infection in this group of healthy cats.
Nope, not in this study.
That's interesting. Did they compare their results to any other research that had been done in Thailand?
They did,
Huh.
and they found that other studies, which often included sick cats,
Right.
they tended to report higher rates of both FIV and FeLV,
Okay.
and they said that maybe it's because their study only looked at healthy cats.
Right.
And
Yeah.
also maybe because the way people manage cat populations in cities like Bangkok has changed over time.
That makes sense.
Yeah.
It's important to remember what these tests are actually detecting, right? Like the
Mhm.
FLV test in this study was looking for the antigen.
Exactly. So that FeLV antigen test,
Yeah.
it usually means the virus is actively replicating, which is what we call a progressive infection.
Okay.
But cats that have a regressive or abortive infection,
Mm-hmm.
where their immune system is keeping the virus in check or it's going completely,
Right.
they would probably test negative for the antigen even if they had been exposed to the virus before.
So the true prevalence of FeLV is likely higher than what this study found.
Yeah, probably.
What about vaccines for FIV and FeLV in Thailand? Are those being used?
They said that vaccines are available, but they're not being used as much as they could be.
Oh, that's too bad. So as we start to wrap things up, what are the biggest takeaways from all this information?
Well, we learned about a really promising new way to classify FeLV using genetics.
Right.
And it really seems like this sharing of genetic material with those endogenous retroviruses.
Right.
It's a big deal in how the virus evolves.
Okay.
And we also looked at that really sad case study that showed just how serious FeLV-related diseases can be.
Absolutely.
And the data from Thailand gave us a good look at how common FeLV and FeLV are in healthy cats in that part of the world.
Right.
And it showed that the risk of exposure can be different in different regions.
Okay, so lots to think about.
Yeah.
What were some of those aha moments for you as we went through all this?
So for me, the fact that the genetic grouping doesn't match up perfectly with the traditional classification,
Yeah.
it really makes you wonder about the biology of these different FeLV variants.
It's like we're seeing it in a whole new light.
Yeah, and that case report, even though it was sad, it was a powerful reminder of how FeLV can be linked to those aggressive cancers.
Right.
And the data from Thailand, it shows us that these viruses are out there, even in cats that look healthy.
Yeah, for sure. So what about for our listeners? What are some things they can keep thinking about?
Well, think about what this new genetic classification system might mean for diagnosing and treating FeLV in the future. And how might geography affect how the virus evolves and spreads around the world?
Yeah, that's a great point.
And the findings from Thailand, they remind us that these viruses are a real concern, even if we don't always see them.
Yeah, that's a good point.
Yeah.
So as we wrap up this deep dive, here's a final thought. Given how much retroviruses like FeLV can change with their mutations and recombination,
Right.
how many more variations are out there that we haven't discovered yet?
That's a good question.
And what could they mean for cats all over the world? This is a really active area of research, and hopefully you have a much better understanding of it now.
I hope so, too.
Thanks for joining us for this deep dive.
My pleasure.